Here we go. All right. So first up, we have BioFile Finder. This is the splash page that you would land on. It's all of our things are located under the subdomain of Balancel. So this one's at bff.balancel.org. And you would be able to go here now, and it's gonna present you with this sort of like thumbnail description that or video that's gonna go over everything we talked about today for BFF. And then there's some descriptions here of various features, sort of like the highest level like impact ones we think, as well as some prompts for how you would like to start interacting with BFF. So there's one for how you would like or getting your data in here. So this will prompt you to load in a CSV or other kind of data source that we'll go over what that looks like later, as well as some open source data sets. And that will link you to these two pages up here. So then we have a learn page, which has some more details if you have more things you'd like to learn about BFF, how it works, some FAQ, and generally like why you might want to use it, as well as an open source datasets page. And this is just a quick little table of all of our published datasets formatted to be compatible with BFF, as well as two from the Allen Institute for Brain Science and one from the Allen Institute for Neural Dynamics. If you have a dataset publicly available you'd like to include in here, let us know. We can totally look into that. So how it looks is once you, in case you didn't catch that, I went up here, clicked this little get started button. This opens the application. And then in the middle here, there's a prompt for choosing a data source. On the left, there's some stuff that's kind of blank right now, but it's just giving you some options for querying. And on the right, this is also blank. So if we choose a data source, which for the sake of this, we can just go to our open source data sets, and you can see this list. You can just choose any of them, but I chose the this variance paper data set here. And now it has rendered that. And what it has done here in the middle is given me that spreadsheet rendered as a file list. So that CSV then just gets converted into essentially another table format right in here. And it's chosen 4 columns from that to show. And if I click on a file, it'll show me the entire, all the details for that individual row, as well as render a thumbnail if it can. So if we go on the left side here, now we see that there's some options enabled, and I can group by, filter, or sort. What it means to group by is I can select something like, say, gene, and then it will automatically render folders for all of my individual files. And so if I click onto one, then I can see that this file says it has this gene that with this folder here. So what it's doing is creating folders that will shove all the files for that particular gene into here. If you have a file that falls into multiple genes, it'll put them into multiple folders. And if I want to create more of a dynamic hierarchy, I can do that. So I can add colony position underneath that. And I can see that I actually have 2 different descriptions for colony position. So it kind of like highlights in a way at a high level some ways that you could do some more harmonization to your data. And I can add more. So if now I have 3 levels. So what this is doing is saying that the first level here are all gene values, and then we have colony position values and then structure. And I can reorganize these on the fly. So now structure's at the top. And then if I open this, it'll show what genes are available for files that have this structure. And underneath that are all the colony positions. So then I can remove them. And again, if I then check this out, it'll say that I have that gene that matches this, as well as the structure that matches the folder. So those are generally how queries work. You can also add filters, which behave somewhat similarly. So if I have a filter and I want to show down just to this gene here, I can do that. And now it's just rendering the folders for the structure and gene that pertain to this specific gene. And I can change it up. I can add multiple. So now it's rendering one of these 3 different genes. And it looks like for the structures, yeah, actin filaments and centrosome, we only have, let's see. There we go. Okay, yeah. So we have 3 different structures available for those 3 genes. And then if I go back here, I can check out some interaction options. So if you have a file selected, you can interact with it in various ways. So one of the ways is I can open the file, and what it's going to do is show some options for viewing. And these can be customized. You can narrow down to whatever you'd like, or have it show about to a specific link. But one of the options I'll show right now. And this is one of the tools that we'll be showing right after this is linking to Volley. So this is a volumetric viewer, and it's going to load that file right in your browser without having to download it. And you can check out that individual file. I won't go too much more into Volley since we'll get into that, but that is just showing one viewer option. There are others as well. And you can provide other interactions. So this would download it. This would download that file into my system. I can save these selected rows out as a sub data set. So if I click on this, it's going to prompt me to select which columns I would like to download because I've already selected my rows, right? So now I've selected my rows and my columns. And then if I open this up, which probably will take forever because Excel is slow. Oh, okay, got numbers. We have those rows here, and we can have all the file or the columns that I've selected, which is all of them. And if you're curious what those data sets look like, this is what they look like. They are just CSVs. So if you want to make one, you can just go into Excel at any time and populate it with any columns you would like, as well as a column called file path. That's the only one that we require. And the reason for that is that we need some sort of reference to a file somewhere. The file can be on the cloud, it can be on your local system, it can be in a hard drive that's totally unconnected to your computer. Where it is connected is only relevant to the things that we're viewing or interacting it with. So if you do have it in a hard drive in a closet somewhere, totally unconnected to your computer, you won't be able to view it, but you would be able to, view the metadata, which is what BFF displays in here, because that is what is in your data set. So it's a way of sort of cataloging your data regardless of actually being able to interact with it. So you don't have to rely on your folder structure within your own file system. So similarly, there's other options. You can, we're recently prototyping this feature called processing files. And what this is doing is providing you some scripts that'll be pre-generated for the files that I've selected. And so if I take one of these, for example, this one says extract standard metadata. So it's going to give you some Python setup. So whatever I need to install in order to run this, as well as the script that's being automatically generated for those files. And if I ran this, it would give me, let's see, oh yeah, a CSV that is all of the standard metadata that we could pull out using BioIO. And what standard metadata means is specific to BioIO. So the tool will explain to you what exactly that is meaning to us. But for the purpose of this, it really just means like there's like 8 columns that we think are generally high value to most people. And we pull those out of the files automatically. And you're also able to edit this. So if I wanted to take colony position and replace it with, I don't know, unknown, I can do that. And then if I go to colony position now, I can see that unknown is now a value. And if I wanted to save this out, I could save my entire query result, and it will give me that same prompt of downloading as a CSV, and this will save that data set back out to my computer. That's important to know because this won't go and interact with your data source where it is. So it's not actually editing this CSV, it is editing what is displayed in your browser. So it won't go into Google Sheets or wherever you have it saved. So your data is totally unedited in its permanent storage anyway. Now, if we go into something like, let's see, I guess we could do numbers. So if we go back out to this one and we look at our little CSV, so if you had data you wanted to show in here, or any sort of table format like this one, you could create a simple CSV. So all that looks like is doing is say we delete everything except for file path here, delete columns. All right, so now we have some file path. I can say like some file somewhere.txt, and then maybe I have a file from my viewing here. So I can steal this file path. This is representing some actual data. Then we can paste that in here. And so these represent 2 different files somewhere. I can add another column. It can be truly anything. So I can say like color, blue, green. And now I have a data set. So this data set is just 2 files, but you can have obviously more complex ones like the one you just saw. And if I want to export this as a CSV, let's see, include table names, blah, blah, blah. Then I can go back into BFF, choose a new data source. So what I did was I went up here, I clicked add, new data source. If you wanted to just do that from the very beginning, what it looks like is clicking that getting started button and choosing a data source. And then it's going to prompt you for something. It'll either prompt you for a cloud-based one. So this would be on like a S3 in AWS or Google Cloud or something like that. Or you can even use Google Sheets to publish your data. If you have questions about that, there should be information in the learn, or we can, uh, message us. Um, but choosing one off your system looks like this. So it'll prompt you. I can choose that CSV. I can click load. And we were not cursed with demo luck today, so it worked. And you can see that it's loading in those two files. One of them has a little thumbnail, right? So this is the one we're automatically generating based off of this file path. And then the other one, yeah. So, and then if we, take this metadata, we want to group them. We have green, blue, just like we selected, and I can interact with them. And then if I want to, actually look at it in a viewer, I can do that. And that lets us do pretty much everything. So that is my demonstration of BFF very quickly. Do we have any questions? See, I can't actually see questions, so I'm assuming there are none. If there were none posted. Okay. In that case, I'll hand it over to Cameron. He's going to talk about Volley.
All right. I'll steal screen share then here. Okay. Let's make sure this is out of my way here. There we go. All right. So Sean already showed off Volley in the context of BFF. Volley is also its own standalone application that is just accessible at volley.allencell.org. And Volley is a 3D image viewer emphasizing access, both in the sense that it's on the web, you don't need to download anything, and in the sense that we've put some work into really finessing the interface to make it friendly, rendering quality, especially with 3D data and with multi-channel data, and emphasizing loading speed as well. So I'm going to load an image with this load button. It's going to tell me that this is primarily optimized for OMI-ZAR images. If you're not familiar with what that means, ZAR is a data format that lets you split your image or any data really into many different chunks, which makes it great for serving over a network like we're doing now, because it means that Volley and other applications that access it can choose to only load the chunks that you're actually looking at any given time. And a ZAR can hold any old data. OME-ZAR is an open standard for how to give a ZAR the right structure and metadata to represent a microscope image specifically. So ZAR is the generic data container. OME-ZAR makes it specifically a microscope image. We can also load TIFFs. We prefer OME TIFFs, which is its own open standard, but we'll do our best if that's not there. For instance, if TIFFs generated with ImageJ should work in here. We have this sort of buyer beware up here that we can't do the same clever data slicing and loading with TIFFs that we do with ZAR. So if you try to load very large images into Volley, you may run into trouble faster than you would with a ZAR image. So I'm going to go ahead and load this image. It's got some odd metadata, but I'm going to choose the colors up here. And now I am looking at this volumetric image here. One more thing about data loading, sorry for all the preamble, is that this is, I pasted a URL in here, right? So this is a tool that's meant primarily for publicly hosted data. It's not currently the right tool for images that are local to your own device. So what that ends up meaning, sort of workflow wise, is this is not something that can sort of slot in right after image acquisition. Where it really shines for us is when those images have gone through a bit more of our data pipeline, especially when we're looking to compile these images into the large data sets that the Institute produces, of the kind that BFF is great at browsing. So these two apps really complement each other. That's sort of the use case where our internal scientists use that tool. And this is also part of our sort of um, strategy for making our data accessible both to external collaborators as well as people in education. So now I am looking at this 3D image of human cells growing in an epithelial colony. Um, each one of these channels is sort of given its own color. This first channel here is the cell membrane. I have this blue DNA channel, and then the green for this image is microtubules. Um, and over here is where I can sort of dial in the appearance of each individual channel that was acquired with this image. If I want this to be a really like high contrast magenta so that I can pick out that feature better, I can do that. Um, as well, if I hit this um, settings button, I get access to a lot more levels to dial in the appearance of a channel. If I'm not really familiar with what I'm doing, these preset buttons right at the top are a good place to start. If I do know a little bit more, I can sort of adjust manually based on the histogram here. And if I know even more, just want to get wild with it, I can go to this advanced mode where I can start doing really clever stuff with adding more points. And even if I right click, giving these points individual colors, um, which is not always the most useful for image analysis, but is, for instance, how we generated the very sort of handsome cell image on the landing page, um, is by dialing in that advanced mode. So let's say I want to sort of move this to a level where the microtubules are visible but not in the way. Um, I down here also have access to these clipping controls, so I can dial in like um, maybe I want to cut away at the volume in order to see what's inside or to see really clearly the sort of plane of these nuclei. And I can also down here start playing through time. This is a time series image. You'll notice, um, first of all, that the image has just gotten grainier when I did that. That's because Czar lets you generate a bunch of, in addition to chunking up your volume, will let you generate lots of different downsampled scale levels for apps that to make a trade-off between data size and image quality. So Volley just now made the choice to sacrifice image quality for loading speed. And then also that first playthrough was a lot slower than subsequent ones because now all of those individual time steps should be cached by Volley, and now we'll play through time a lot faster, and you can see these two cells undergoing mitosis and those single nuclei dividing into two nuclei with the help of the microtubules there. Um, and then if I let this settle down on a time step, let's say right before both of those cells are about to split, I get my higher resolution level back. Um, More rendering settings up here. I can pop into 2D mode. Continuing on the theme of adaptive resolution, I've now just gotten a higher resolution image because, again, this is chunked. And now that we're looking at just a single plane of the image rather than the entire 3D Z stack, we have more budget for more data in your browser. Um, this is also sort of a common misconception or like question that we get when we show off this tool is that's great, but does it work for my 2D data? Yes, it does. We have external collaborators using it for 2D data. I can show off this image from Cell Painting Gallery, which, you know, the natural default is for it to go into XY mode. All you get with a 2D image in Volley is this sort of like trivial case of volume rendering where you can rotate around, but you don't see any depth because there's none there. Um, so I also up here have a couple of controls for showing this little axis indicator if I'm lost in 3D space, as well as this bounding box. The bounding box is really useful for getting a sense of scale because now I have these tick marks along the edge, along with an indicator for how far apart those tick marks are spaced. And then the big thing up here is this render mode dropdown, where I can pick between different ways to render this data set. Volumetric is sort of the default. I can also go into max project, which is a mode that sort of loses a lot of depth information and instead sort of like gives you this transparent view at whatever is most intense through any given perspective of the volume. So now you can see, for instance, where these microtubules extend into the chromosomes as they're splitting apart. And the final mode in here is this path trace mode, which is going to take a second to load. And then that actually already looks kind of reasonable, but I'm going to go over here to these advanced settings where I have a few more controls for how this looks. This mode tends to look best if you dial up the density and brightness a little bit. And then you can see it sort of progressively rendering in. This is a much more physically based mode where it's actually like throwing light rays into the volume, simulated light rays into the volume, and then seeing how it reaches back to the to the camera. This is kind of a miniature version of the rendering technique that AGAVE is really committed to specifically. You'll learn about AGAVE much later in this talk from Dan. You can kind of see in this render that, for instance, we're getting some shadows here where there's sort of a cranny between these microtubules and this bundle of DNA. And then the final thing I want to show off for today, besides, let's see, I have the option here to take a screenshot once I've really dialed in these settings and let the like samples of this rendering algorithm accumulate. Can download a screenshot if I want. As well, let's say go back to volumetric, and maybe I've dialed in some setting exactly how I want in order to reveal some interesting feature of the volume. We have this share button, which will copy you out a URL. And not only will this URL take you back to the same image, it should take me right back to the same view configuration. So you can, if you're looking at an image in Volley and want to share the exact same perspective you just dialed in with a colleague, you can do that with this share button. Um, then spinal buttons, I can reset the camera and give my volume a little turntable effect. Um, that's about it for what Volley can do currently. I'll end by sort of describing the features we have upcoming where we want to take the viewer next. One is a widget for specific control over um, what scale levels you're loading out of a czar. Currently, Volley is making a lot of decisions for you as to what scale level to load based on the mode you're in and the data budget it thinks it has within your browser. Um, but we're working on giving users a lot more control over seeing what scale levels are available to them and choosing another one. As well, we are working on better integration with BFF. Obviously, BFF can already pop out an individual volume or an individual image out of a row of that spreadsheet view. We also want to set it up so that you can make a selection and open the whole thing in Volley with options to um, navigate between those images as different scenes, and as well to drop any additional uh, metadata that BFF might know about into this metadata viewer over here, which I didn't show off before, but we can accept sort of arbitrary metadata in addition to um, what is already provided by the OME standard. So I think that is it for Volley, unless my colleagues think I missed anything.
There was a question in the chat earlier um, from Vid Sharma. So does it work for a large whole slide of 2D images, like tens of gigabytes? Um, I added a response to this, but I'm curious if your response is going to differ at all.
Um, I I mean, depending on I expect it would, depending on the size of each individual image. And in particular, like, I guess as a data prep question, it looks like that's sort of the theme that Dan had in his response. Um, that, oh, yeah. So it's probably mostly a data prep question of um, like original size of a data set doesn't necessarily tell you much and without like how is it split up into different time series or scenes and how much are you willing or not to um, generate like scale levels downsampled from the original or um, how much memory are you willing to let your browser take up because there's um, we are currently a little bit conservative. Again, we're giving people controls over that sort of thing. But um, there's yeah, once those controls are in, you'll be able to um, do kind of whatever you want with your own computer's memory. All right, I think I have um, that's sort of the time that is allotted for me. So
Right.
take it away, I think, Peyton.
Yeah, I also am curious if because we've mentioned OMEZAR before, we also do have a Python library called BioIO that you can use for converting between OME-TIFF, like opening and converting between different file formats. I'm going to link that here in the chat as well. And so this works like you can basically install plugins if you're like, I want to open a certain type of image or I want to save as a certain format. And that makes it easier to do things like saving your images as OMIZARs. Okay, so I'm going to go ahead and get my screen sharing set up. Um, just as an overview, I think we linked to the document earlier in the chat for anyone who's new coming in now. I'm just going to share this again. Um, there is a link to sort of our overall outline. I'm going to be talking about our last tool of the first sort of half of our presentation today, and then we'll be going for like a quick break for about 10 minutes, and we'll come back for a second half after that. Okay, can everyone see this? Perfect. So I'll be talking about first Cell Feature Explorer today, and later we'll also be getting into Timelapse Feature Explorer, but we'll start here. So Cell Feature Explorer is another tool as part of our overall suite of visualization tools. And what it is really specialized for is traversing large data sets where you have like lots and lots of images. You're interested in exploring maybe relationships between them and different features, and trying to understand more of like what's happening for these really large data sets where there's lots of images. So here we have a couple of data sets that we've published on our landing page. And also, I'm going to go ahead and stick this in the chat too, if I can figure out where the chat is. Um, here you go. Um, so Cell Feature Explorer has a couple of our existing data sets that we published. These are from our variance data set, and there's a few other ones from like some of our like other HIPSC specific ones like disease cells, drug perturbation data sets. But for now, I'm just going to go ahead and load this. You can see that there's 215,000 cells in this. Um, And once Cell Feature Explorer has a second to load all of this data, this may take a little bit, we're shown this really lovely plot view. So here in the plot, if you hover over each of these points, each of these represents one of our images. And over on the left, we also have currently we're coloring by the labeled structure in each of these cells. So this is like everything like actin, we have centrosomes, the ER, nuclear envelopes, etc. I can color by not only like this labeled structure, but also features that we've measured for each of these cells. So if I want to look at like cell height, for example, or cell volume, I can color all of that on this main plot. I'm going to switch back to cell structure because I just think it looks nice. Um, I can find it again. There we go. So over here on this main view, this is a way of sort of exploring, again, those features that I have in my data. So right now I'm looking at DNA volume and DNA and cell volume. So there's some sort of like ratio between the nucleus and the cell itself. And you can see here just from this graph, we can already see some patterns in our data. So there's sort of two lobes where one is sort of following this very linear relationship between cell volume and DNA volume. And in this other one, the DNA volume is actually much lower compared to our cell volume. And so maybe I'm interested in that. And I can actually sample a few cells out of here by clicking. You'll see that they're going to update on the right side in this thumbnail. And if I start looking at some of these cells, you'll notice that we've actually popped over into Volley, which is integrated into CFE. So you have all of those really nice volume controls. So if I split here, we can actually see that this DNA is really, it's starting to condense. And if I look across all of these cells actually that I've selected, which are all in that lower lobe, let me see if that will load. This one also has condensed DNA, and this one as well. And so from that, we can actually conclude that this branch here, or this lobe of this data, is actually representing cells that are in the middle of mitosis. And so the DNA is condensed. It's a lower volume relative to the overall cell volume. From this plot, I can also zoom in. So I'm clicking and dragging here to look at a smaller view. Can do that again if I want to. And any of these can just be selected and popped open in the side view. So here, for example, this is, I believe, the nucleolus. Um, and again, I can tune sort of any of these visualizations here with these controls on the right side to try and dial in my image. I can also slice into it to see exactly like a certain plane, or I can switch to a 2D mode and view just the like a 2D plane of that data. So this is great if you're looking at our data sets, but what if you're interested in data from BioFile Finder or your own data sets as well? We have some features that are currently in progress. We've been working on these internally, and we're excited to get these out, hopefully in the next month. But from BioFile Finder, so I'm going to open up sort of one of our internal data sets. So this is all internal data. Um, I'm going to load this in. I'm going to set a filter for just OME SAR images. So I'm going to, oops, search for kind, and then I'm going to search for OME SAR. This gives me a list of, I think these are like 5,000 images, and I'm going to select like a big healthy slice out of this. So this is 3,400 images. I can right click, and then here there's an option to save metadata as a CSV. So I'm going to go ahead and download that. I'm going to select all of this just to keep things nice. So all of this metadata is coming along for the ride. So download. BFF is going to take a second to just process that and turn that into a nice CSV for me. And then over here, this is again an experimental early preview version of Cell Feature Explorer. If I click over here, oh, I'm going to zoom out one. This is a little zoomed in. If I click over here in this top right corner, there's a load button, and I can use this to actually upload either a CSV that lives somewhere on the web or a CSV on my device. So in this case, I'm going to go ahead and load this file here. This is the CSV manifest that I just exported from uh, BFF, and I'm going to pick, let's see, I think it was well label and position index, so you can get a better view of this data. It can be a little small otherwise. So let's see. There we go. And so you can see that this now has some of those representative images in here, and I can actually click into these. If I hover over them for long enough, we're actually dynamically generating thumbnails based on that data. And so if I go over here, I can now preview any of those files in Volley. And so this becomes a way that if you have a large number of two-dimensional images and you're really interested in exploring them, or even three-dimensional volumetric data, you can do all of that from this really nice plot view. And again, all of the features that are here, these all came from like metadata columns in CFE, which means that you can upload like any custom data, any custom like column to associate with these. So as another example, this is also sort of internal data. I can load, this is another view of the variance data set, and we're doing some like principal component analysis on it. So I could, for example, visualize by cell structure our whole like PCA map. And then if I'm like, oh, I'm really interested in like what's happening in this corner, I can click into any of these cells to visualize them. So here, for example, this is, I think, just a crop of one of the images in the variance data set. We'll see if this loads or if I'm going to get demo luck. I'll try another image. Let's see if this one works. Oh, that one might have just loaded. There we go. So that's like a smaller crop of the whole cell. And there's the whole volume. One thing I also forgot to mention too, and this is a feature that CFE uses that's related to what Volley can do, is over here in our variance paper dataset, where this is like, we're currently looking at sort of like blocks of cells. You can either switch to a full field view to see like the whole uh, capture that included this cell. And then also back in this view, I can mask out everything except for the cell of interest. And these are like you can sort of put in specific channel names in order to set up like this masking. And you also have support for doing things like including like a more like larger context crop. Okay, that was a brief overview of CFE and some of the features that it provides, as well as a quick overview of how it integrates with BioFile Finder. Are there any questions? Okay. Um, we'll still be open for questions if there are any. For now, we're going to go ahead and take a 10-minute break. And I'll still be online, so if you have any questions, we can address them when we come back. But we'll see everyone in about 10 minutes. So intimidating coming back to a Zoom window and everyone has their cameras off and is muted.
I'm still here.
Yeah, how's the conference going for everyone? I was looking online and I was pretty interested in some of the other talks, but a lot of them were also happening at the same time, and I'm like, oh,
Yeah, I was just looking through those right now. I'm having the same issue.
yeah, I'm really glad that they gave us like a big block because we we have a lot of tools, but oof, I want to see everything.
They are all recorded.
True. I feel like I'm so bad about actually going back and watching recordings when they happen. There's always something else to be working on, so I'm like, ah, do I really want to slow down and watch that? Skill issue.
So, Peyton, I have a question.
Yeah.
Um, if I liked the functionality of Cell Feature Explorer and wanted to try it with my data, What would you suggest is sort of the easiest path to get my data in there?
The easiest path to get your data in there. Um, there's like a man, I have documentation that I've written that we haven't put on CFE officially that I'm realizing I'd really like to. Um, right now, so we're using like if it's saved in BioFile Finder, like any image, any CSV that's saved out of there, we like sort of know because BFF has reserved columns. Um, there are a few specific columns that CFE will look for when it's trying to parse your images. I believe off the top of my head, and if you are interested, please email me. I'm gonna stick my email in the chat. Um, and this will be good pressure for me to remember to make documentation for this once we make the CSV feature live. Um, basically, you can keep your CSV as long as you have a URL to your image. I believe we call that like the file path, but I'm going to check really quick just to make sure I'm not giving incorrect information. Everything else, like CFE will automatically parse into a feature, or if it recognizes that it's like metadata from file Finder, it just won't show it on the UI.
Okay, so it sounds like what you're saying is just make a data table, like basically any sort of spreadsheet type of data.
Any sort of CSV format should work, and then you just need to make sure you have one column that represents your images.
Yeah.
And those should be URLs. I'm gonna really quickly check, where is this? Yeah, the one thing that we're looking for is file path. And that will be that HTTPS URL.
And for Cell Feature Explorer purposes, that's the thing that links a plotted data point to an actual image.
Yes.
Yeah.
And we have about 2 minutes left in our break. I'm very excited for this next section. Any other questions from the rest of the audience? If you have anything, feel free to put it in the chat or unmute. Happy to take any questions. Hmm. Okay, I think we were right about a time. So just as an overview of the second half of this talk, we're going to be talking about three additional viewers. The first one is Timelapse Feature Explorer. I'm going to be presenting on that. The next will be Simularium, and then which is our simulation viewer, and then AGAVE, which is our GPU powered like desktop renderer. We have time also booked at the end for any questions, but again, if you have any questions throughout, like feel free to pop them in the chat, and we'll either respond there or use that dedicated time. Okay, so in this next section, we're going to talk about Timelapse Feature Explorer, and I wanted to start by talking a little bit about what originally motivated this project. So, we, as part of like one of our projects, which eventually led to a paper studying nuclear morphogenesis, we were wanting to like create videos where we could visualize both where our cell nuclei were in these sort of big uh, time-lapse microscopy images that we were capturing, as well as like coloring by certain features that we were measuring. So for example, if we wanted to color by height, we could use the segmentations that we've made of our cell nuclei and then color those images uh, with the color map according to that feature height. So that ended up with videos like this, which are really nice because you can see like at the center, we're sort of starting with this like domed, like higher, darker heights in the center. And over time, that's sort of flattening out. And you can even start to see that pattern inverse where we end up with like this sh like shorter cells in the center of the colony. But every time a scientist wanted to change this, it would take like these videos were taking like 3 to 5 hours to generate. And if you found a mistake or you wanted to adjust a segmentation, then you had to redo the whole thing from scratch. Um, and so that was like a huge bottleneck for our workflow. We were like, these videos are awesome, but like also we have to start running them overnight. And so we asked ourselves, is there a better way that we could be doing this? So that idea eventually snowballed into Timelapse Feature Explorer, and then as we kept working on it, first it started as this very simple coloring tool, expanded more and more for really robust data analysis of segmented time-lapse data sets. So I'm going to switch over to Timelapse Feature Explorer itself. If you want to follow along on this section, I'm also going to figure out where the chat is and stick this in here. So Timelapse Feature Explorer, again, is our viewer for segmented time-lapse data. So that's anything where you've taken like a time-lapse series of images, you've said, okay, this is one object, this is another object, this is another object, and then now you want to visualize that. So here we have a data set that we released as part of a May 2025 publication, which is again about that nuclear dynamics within colonies. I'm going to go ahead and click this button, which will open one of our data sets. And we can start to explore this a little bit. So this data set right now, we're looking at a feature that was provided by the data set itself. In this case, it's volume. None of these are sort of pre-generated by TFE. Like some of our other tools like CFE, this is all user-provided data. Right now, we're looking at volume, which we can actually open definitions that were provided by this data set. So for example, volume is the sum of the voxels inside of each of these nuclear segmentations. We can also look at things like height, which is here in micrometers. It's the distance between the top and the bottom of our nucleus segmentation. From there, right now, I have this color map assigned, but I can adjust this on the fly and change sort of the contrast settings of where we're applying this color map as well. So if I want to like sort of make values at the upper range darker, I can adjust this here. And from there, I can actually just start playing through time. So if I play through this right now, you can see all of these cell nuclei moving. But we also have a lot of other additional settings. So for example, if I want to show the context of where these images were actually acquired from, I have that as a backdrop. So this is the original like fluorescence microscopy image that was captured. It's imaging on lemon B1. So if I wanted to like adjust the opacity so we're just seeing that image, I could do that. It'll be a little bit slower because it's loading in this whole image. And I can also adjust object opacity on the fly. And then I just kind of showed that really quick, but didn't stop to examine it. You can also zoom in on your images, pan around to look at different parts of your segmentation data set, or go back to sort of that main reset view. Okay, so just this view is not super exciting. And this is where TFE started to add in more functionality. So if I'm interested in like what one particular cell is doing, I can actually click on it on the UI and it'll pop up, it'll select that whole track. So everything in that entire object's lifespan, I can now look at. This is showing me this path, which will update as it moves. I'm going to turn off the backdrop so things load just a little bit faster. And over here on the right, I'm seeing the currently selected feature, which in this case is volume, plotted against time. If I'm interested in any particular point in here, I can also click directly on this plot to jump to that moment in time and see what that cell is doing. This also works if I update to a different feature. So right now we're looking at volume. I can also visualize on height. And you can see how the height adjusts or changes over time. And I think there's like 40 something features in this data set that all you can compare between, you can plot over and look at. What's really useful too is that this allows you to associate like phenomena that are happening on screen or sort of like in the context of your data itself with your numeric feature data. So if I look at volume, I'm going to just hunt around and see if I can find a good example of this. For example, in some cases, there are sort of these strange dips in volume, and which is a little bit odd because the rest of this trajectory is so linear. So if I actually jump to that point, let me see if what I think is happening is happening. Maybe not for this one, but I'm going to look for another example. Um, in some cases, let me see if this is happening here. Maybe a little bit. In some cases, like there's sort of like questions that you can ask about your data and associate it back with the original images. So here, like there's another sort of very localized dip in volume, and it's more noticeable on other cells. And so from just a data analysis perspective, I might notice that, but not really know why that's happening. But with TFE, I can look closely at my data and see, oh, at this moment in time, actually there's another cell nearby that's dividing, and that's potentially putting pressure on other cells nearby and causing that nuclear volume to briefly dip. You can also look at sort of broader views across your entire data set. So if I clear my track here, this is plotting the volume for all cells at all times. And I can also plot this against other features in my data. So if I want to look at height against volume, for example, I can do that. Um, well, there's a couple really interesting ones that I'm going to find. Let's see, time, filter by this. Synchronized time, for example, by volume. You can sort of see like the trajectory of each individual cell nuclei volume. And if I'm interested in like a particular outlier, I can just click on that to see exactly what that particular cell was doing, or if there's something strange about its trajectory versus others. So that's a quick overview of some of the like data analysis features of TFE. The other thing I'm really excited to talk about is annotation. So within TFE, because it's such a visual viewer, we also support creating and labeling objects. So here I'm going to go ahead and create an annotation. In this case, I could call this, let's say like segmentation errors. This is for the use case where maybe I'm trying to like note that there are issues with uh, segmentations and I want to go back and adjust it. In this case, this data set looks pretty good, but just for the sake of this. So I can select like basically click on this UI to label each of these individual cells. And then if I go to a different frame, I can label some more of these. I can also select an entire track at once. So if I want to label everything in this track, um, I can do that here. And I can also make multiple annotations at the same time. So if I want to make another one, maybe this time I'll call this cell type. I can click here and then click again to edit the type of the cell. So I'll call this like type A. This one could be type B. And then each time I click, I'm labeling that entire cell track. So if I then play through time, you can see that those labels are going to persist and follow those cells. And then if I want to then update my original data, I can click Export CSV here. This will save out a CSV file that I can then use to it'll have like the label and track number, and I can use that with Python to programmatically update my actual underlying data. And then the last thing I'll show here too, I'll turn off annotations. If I have a view of TFE that I really like the other viewers that we have, they support CS or URL sharing. So if I copy this URL here, which I've just done, and I open like an incognito window, if I paste this in, I'll see the exact same view as my collaborator. And this is a really nice way to send like stable links if you're interested in like sharing data views on data. And with annotations, you can also export those and import them into a new tab or new instance of the viewer. I also wanted to show, right now we've just been looking at two-dimensional data, but TFE does support have experimental support for three-dimensional data as well. So this is the same set of segmentations from the data set where these are the original 3D segmentations that we flattened down to a 2D image. So if I play through time, this is going to be a little slow, but this is also using Volley under the hood, so we get really nice like playback and dynamic like resolution loading. So you can see here that my cells are working and growing. I can also click on them to see a path. There's some issues with the data here, which is my fault. I can also visualize on different volumes. I can change my color map, and all of the settings that we've seen earlier in TFE still work here. You can also do annotations in 3D. So if I wanted to annotate this with like a segmentation label, that'll also work. It's a little bit odd because of some, again, Centroid issues that are my fault. I think I have one more data set that I can load this is all like experimental data, so these volumes are not super clean. Oop, never mind. Demo luck. Okay. So that was a lot of talk about the viewer. And if you all have any questions about the viewer, we can slow down for that. The other thing I wanted to show is how to actually get data into TFE. So from TFE, if you go to our help button, there's a link to our GitHub repository here. If you scroll down on this, there will be a link here to a getting started guide. And this is a Python notebook that you can run to like basically go through a test workflow of how you actually set up data for TFE. This button here opens in Google Colab, which means that you don't even need to install anything to run the demo. And I can just really quickly talk through this while I have some of my prerequisites installing in the background. So I'm going to get these two started. This all works with a companion like data preparation package called Colorize Your Data that we released along with TFE. So this is really expecting segmentation images that are going to look like this. It's any sort of image where you have numeric values that are assigned to pixels that represent the object in question. So in this case, this object has segmentation ID 1, this has 2, and this has 3. Then for like time-lapse data, you can link these all together over time across tracks. So here, like this track number 5 has different segmentation IDs across different frames, but we know that this is all like the same cell. Then for your data itself, that's typically going to look like a CSV file here. So you want to include like a segmentation. Oh, that's not what I wanted to do. How do I close the editor thing? I think I can just run this. That's also not what I wanted to do. There we go. Here you're including like a path to your image file, as well as a little bit of additional metadata like your track ID, your time, and some Centroid information. Everything else is going to be automatically parsed as a feature. So in this test data set, I have area, radius, and location, which is like either bottom, middle, or top. And those will automatically be parsed by TFE or by Colorize Your Data into a data set that TFE can load. So I think I accidentally ran every single image, every single thing in here. I'm still waiting on some requirements to install, so we'll give that just a minute. Once you're actually ready to convert your data set, it's just one API call here, and I'm specifying some of the columns out of my CSV data set that I want to load. But once that's done, I have a functional data set that I can load in TFE. These additional steps are sort of setting up extra metadata to make things look extra fancy in TFE, but all of this is optional. And I believe, let me see, down here, oops, this is not what I wanted to run. This is, as part of this, going to create a zip file, so I can just run this again, that then I can load into TFE. So if I go back to Timelapse Feature Explorer and click load, I can load a zip file. And this is the file that we just pulled out of Colab that then has this converted data set in it. And you can see that this just loads. I can switch between different data sets in here. I can switch between different features. I can click to like plot a certain feature along this, and all of that just works. If you're running on a local machine, we also have a command TFE open that can just automatically open like a local file for you. So I believe all of these ran, and then I can just do this. And this will open in a new tab TFE with my data set already loaded for me. Yeah, that was a very fast overview of TFE and some of our tutorial resources. I'm going to link this getting started guide as well in case people are interested. Um, I have definitely gone over my time. Does anyone have any questions? Okay, if not, then I think we can go ahead to the next part of our talk. So I'll hand it over to Dan.
Thanks. Let me screen share. It is not allowing me to share right now. Sean is nodding.
Classic.
Is it like a
Sorry.
restart
Zoom
now?
is showing me just like exclamation marks on all of the thumbnails for sharing.
Hmm.
I can try to share anyway, but let's see. Ah,
I just clicked a button over your face that said make co-host, so I don't know if that's changed your permissions at all.
I believe it's going to make me rejoin.
Oh boy. Wouldn't be a demo without some technical difficulties.
Let me just see if I don't rejoin. Oh, wait, I can do it.
Hooray.
This looks reasonable. Can you see slides?
I can.
Yes?
Yes.
Okay, great. Okay, so I made a few slides just to help give context before I do my demo. So at this point, we are up to two more viewers to show, or two more tools. I'm going to show, these are two very different tools. One of them is Simularium Viewer, which is for sharing simulation trajectory results for the modeling community. And then the other is AGAVE, which is a sort of advanced volume rendering and visualization tool, similar to Volley, but sort of designed for a higher end result for image generation. And all of the tools that we're talking about today are, you can get to them via just going straight to allencell.org. And then at the very top of the screen at that website, there's a data and digital tools menu. And it has links to, I believe, all of these. So getting into Simularium, this tool has been created to solve problems in the modeling and simulation community. In particular, the tools for creating simulations tend to be highly technical, lots of coding involved, and the individual simulation engines that people use are either homemade or there are a few like known ones, but they're generally hard to use and they're all very different. And then once you've run a simulation, it can be really difficult to visualize and share results. So the goals of the Simularium project are to remove some of these challenges and make it more accessible to people who may not have the technical background or skills. So we want to make it possible for wet lab biologists to use simulations and visualize results and share results. Collaboration is a big part of this. So making this a web app means we can share links and easily communicate with people across the world if we want to. Accessibility is another issue. So we spent a lot of work on making this as easy as possible to use so that it can be used in classrooms for educational applications. And yeah, we also have thought hard about how to think about like basically having multi-scale simulation and modeling and be interoperable at multiple scales. So there are, I think this last one is a little bit more of a future concern for Simularium. You won't see anything that specific about multi-scale in this presentation. So, but the main idea here is that we want to do an easy to use interactive presentation of 3D simulation results. And reasons why this is useful, of course, is that with interactive 3D, we can explore spatial relationships. You can play back time series data again, which we've seen in a couple of the other tools, to get a feel for the actual dynamics of the system that's being modeled. And then intuitive navigation of data sets. You can click on individual parts of the simulation and zoom in on them really easily. So I think that is my intro. Yeah, so I'm going to step into a demo now. So again, this is just an overview of the Simularium viewer. We're going to display agent-based simulation trajectories in the browser. Simularium uses a custom data format. We provide converters from many well-known simulation packages in our Simularium IO package. We also support automatic data conversion as sort of an experimental feature, which I'm going to show in a second. Simularium can display agents of various appearances. In particular, it supports spheres, triangle meshes, PDB, which is protein data bank molecular files, and fibers, which basically are parametric curved shapes. Yeah, and the viewer is implemented with TypeScript, React, and WebGL. So from there, I'm going to, oh, and this is a sampling of the many, several of the different things we've gotten through this viewer. But I will step in and actually show some real ones and some of the features of the viewer now. So when you go to simularium.allencell.org, you get this landing page. So we have several preloaded trajectories here on a server that you can load. These are all associated with real publications, so this is actually proper data. I'm not going to attempt to explain the science in any of these. But let me just pick one of them and go right into the viewer. So I'm just going to take this one, for example. So when you load that simulation into the viewer, so the idea here is that the simulation has already been run and someone has already made sure that the output is in the Simularium format. At that point, it can just sit on a web server and the data can then be streamed from that server to this viewer. So again, we have a 3D viewer, as with some of our other tools. So again, we're kind of going to have to get used to navigating in 3D space. So typically, you just click and drag to start rotating the 3D view. You can scroll up, down to zoom in and out when your mouse is in the 3D view. And you can, I think, shift, drag, or I believe like option. Oh, so no, I'm on a Mac, so I'm using 2-finger click and drag on the trackpad to do panning side to side. So if you can you kind of get used to these become intuitive after a while. Click and drag to rotate, 2-finger drag to pan around, and scroll up and down to zoom in and out. This particular viewer also has some buttons on the side to do that kind of thing. So you can just quickly zoom in and out. It has a default rotate mode. You can actually switch that mode so that the main click and drag does panning if you feel more comfortable using these simple buttons. Okay, so on the left-hand panel of our view, we have a list of all of the agent types in this simulation. Oh yeah, so I haven't really defined what an agent is. For the purpose of the Simularium viewer, an agent is sort of one single thing that is moving or changing in our simulation. So in this case, all of these little yellow spheres are agents, individual agents. And in fact, there are more than just the yellow ones in here. There are these sort of amber colored ones. And apparently, according to my map here, there are 3 other types that I'm not actually seeing right now, which indicates to me that they probably are going to show up later on in the time sequence. So at this point, you can just, so obviously I'm clicking these checkboxes to hide and show agents of a particular type. I can click the star button to highlight all of the agents of that type. So if I hide these and then click the star on the amber color, it's going to just put an outline around them. So I can now bring the other ones back, and you can see inside of there, they're still highlighted. We can also change colors. So you can just recolor these if you want to. When you produce the simulation in the first place, you get to choose these colors. That's pretty much everything you would typically do on this panel. You can also click on individual agents. So I'm just going to hover over one of these and click on it. The view is in a mode where as soon as I click on one, it's going to center the view on that agent, and I can zoom in to get a little closer. And as I click around, it's going to jump to each one so that it's centered. That's a control that you can control with over here in the lower right called focus. If I turn focus off, then I can click and it won't center everything. So I'm going to put focus back on, and it recenters. As soon as I've selected a single agent, I can actually see some basic information about that agent in the lower left. In particular, we can see its position and orientation in the simulation. So again, an agent is a single object that's moving in the simulation, and every agent can have a position and a rotation angle. In this case, these are spheres, so there's no real difference between their rotation angles, none that you can visually tell. So at this point, I'm going to turn everything back on. I'm going to zoom out a little bit, and I'm going to press play, and we'll just watch the time sequence go. So you can see everything changing. We've brought in the new agents of different types. That's about how fast you can play through one of these simulations. The time scale itself, again, is part of the data. So a simulation can run over hundreds of seconds, or it can run all inside of, a nanosecond. It just depends on what you're simulating. This is general against anything. Another interesting aspect of the viewer is that you can also embed plot data with the simulation data. So when you're preparing the data file, it's not just doesn't have to be just a collection of agents and their positions over time. You can also provide, in this case, it's just a count, but you can really provide any plottable data over time. And as we play through the simulation, we have a little time marker on the plot here. It's also the case that you can provide multiple plots. So it's possible to have other boxes on this right-hand panel going down. And they will all update in real time as we play through. So again, some of the interesting things you might do with this is hide different types of agents to just watch the progression in time of a particular agent type. So you can see that the distribution is going from one end of this thing to the other as we play through time. You have a setting here to loop. So just keep playing forever. You can also do 1 frame at a time, advance and rewind. So we can just skip one time step at a time if we want to. There's also a movie recording feature, so you can actually output movies of your time series playthrough. Let's see. So for any trajectory that you've loaded in here, you can download it. It actually just downloaded. So this is about 30 megabytes for this particular one. I think share just creates a link to the viewer. You can also tell it to start at a particular time if you want to share a view of the trajectory at a particular time step. You also can do an embed code, so you can embed the whole viewer into a web page with a HTML iframe if you're so inclined. Let's see. So yeah, I think we try to put something like this into all of our web apps. We have some resources around getting started. We have a forum where you can ask questions about the viewer. It's all open source, so you can get to our GitHub. If you find bugs, you have two ways of submitting those bugs to us. A little information about what version of the viewer we're using, which helps for debugging problems. So you also have a load model menu. And we have all of the example models from the main landing page of the website as quick links here, so I can load a different one. So the model I was just looking at before this was using entirely spheres for its agent representations. This one is actually using PDB molecular geometry. Pretty much all of the smaller molecules here are PDBs from the protein data bank. So these are actual protein structures. And these larger purple objects are actually triangle mesh representations. So we're now mixing two different agent visual representations. Again, internally, all Simularium really knows is that this is an agent at a particular position with a particular orientation in space. And then it's given some kind of link to where the geometric data lives. So there's some triangle mesh file for this one and some PDB file in the protein data bank for this one. Yeah, so what else? So I was going to talk about data conversion. So how do you get your data into this tool? If you've created a trajectory file already that's in the Simularium format, you can actually drag and drop it into the viewer. So if you have a file on your local system, you can simply drag it in here and it will load. That's also represented by Simularium files. So you can also open a file browser on your local machine or bring a web link to the viewer like this. And lastly, this is slightly experimental, but we have other file type here, and this will pop open a form. If you have a file that's already been generated with a particular simulation engine, and in this case, currently we only support the Smolden engine, but we would love to hear from you if you actually have simulation data in a different format, and we can easily add this. But this will let you bring in a Smolden file, set up some parameters, and sort of run the conversion for you automatically. I didn't prepare a Smolden file to convert here, but once you've added a Smolden file, it'll give you a little form to fill out with some of the basic information that we need to do file conversions. Now, if you have Python capabilities, you can take a look at our Simularium IO project. This is our library that lets you convert all manner of trajectory types. These are the spatial trajectories that we support right now. So we've actually implemented converters for Cytosim, MCell, Medyan, Physicell, Ready, Smolden, Spring Salad, and MD Analysis. And again, if you have a custom trajectory, or a custom simulator of your own, then you're probably already writing Python code and you can output to our format. This whole code repository has a variety of tools for generating our Simularium files. So I don't actually know how much time I just took, but that's kind of everything I wanted to show about the viewer. Oops, I'm going to.
I think you're perfectly at time.
Okay, great. So any questions or concerns about Simularium? And then I can move on to Agave, if there aren't any. Take a quick look at the chat if I can. Actually, I can't. I can't see
I'm
the
not
chat.
seeing anything coming
Okay.
through from the chat, but again, if folks have any questions, feel free to drop it.
Yep. Okay.
And we will answer them at the end. Okay,
Yeah.
One more tool to go.
one more tool to go.
Yes. Okay. So Agave is the name of the next tool I'm going to present. This is a desktop application. So this is the only one I think we're showing that's not a web application. You don't access this in your browser. This is a desktop app that you have to install, and it is designed for high quality cinematic GPU rendering of multi-channel volume data. So this is designed to display the same type of data that Volley shows. Um, One of the key differences here is that you can, since it's a desktop native app, you can load data from your local hard disk easily or your local network attached storage. Pretty much anything you have a path to, you can load in here. So it supports the Zeiss microscopy file format called .czi. It supports TIFF files. Again, it supports the OME TIFF flavor, and it also supports TIFFs that are generated from other tools like ImageJ, Fiji, or even Python code that just is using some TIFF writer like TIFF file. It also supports this CCP4 MRC volume format, which is maybe a little bit less used in the Allen Institute, but is another volumetric file format. And it supports OME-ZAR. Of all of these formats, currently ZAR is the only one that you can load directly from cloud URLs. So if you have a public URL to a data set, you can bring in ZAR data. We really like ZAR because of the features of being able to load very large data sets in the chunked format. So it's much more efficient, and it sort of makes it possible to load very large data where maybe it wasn't before with some of these other formats. So Agave features a photorealistic camera and lighting model for visualizing the data. It can load time series, and it has a mode where you can render out image sequences from your time series automatically. Built with C++, uses Qt for the user interface and OpenGL for graphics. We have a homepage on our website that has key links to Agave. I'm going to show that homepage in a second. First, I'm going to just show a very quick sampling of the types of images you can generate from Agave. All of these are done inside of Agave, and then I'm going to do a live demo of it. So these are just some of the images we've generated. These are images that our institute didn't generate, but other users of Agave did. Different types of volumetric data. This one on the left is showing an aspect of the physically based camera model. So you can actually do depth of field blurring in the foreground and background and choose a focus point in the midground if you want to. You can move that focus forward and backward if you like. And these are also showing sort of some of the lighting and shadowing aspects from the physically based lighting model. So it really makes it easy to sort of pop out 3D features in your data. Okay, that's it for slides. Let's take a peek. This is the homepage for Agave. This has links to sort of what's new in the latest version, a quick link to go to the download location, which is at our GitHub site, a link to the sort of main page of the GitHub repository for source code. We have a documentation link and a help link here. That's the main stuff you need to know. And then there's a little bit of information below here about just sort of the philosophy behind this and why this type of rendering can make a difference in visual analysis. So from here, I'm going to launch Agave. And interestingly, one of the ways we can launch Agave is directly from BioFile Finder. So, oh, sorry, let me just quickly show how you would get Agave and install it. So if you click this download link, takes you to our this GitHub page with the latest release. We provide pre-built releases for Windows and Mac OS. Agave does run on Linux, but in order to make it run on Linux, you have to download the code and build it yourself. And we have pretty simple instructions for doing that. I think it's rare that we actually have trouble building it, or I mean that anybody has trouble building it. But of course, if you try that and have problems, you can always let us know and we'll try and get it fixed. But we currently provide pre-built packages for Mac OS and Windows. And so you would download the one of these that's appropriate for you and then proceed from there. So once you have it installed, you can actually get to it through BioFile Finder. So I'm going to load one of our open source data sets. I'm going to just load this particular one. This is our variance data set. I get a list of all of our image files. I can click on one of the files, right click, open with Agave, and it pops open an Agave instance. We get a preloading dialog here. Because this is ZAR data, it's going to let me make a few choices before I actually load the volume data. So in this case, this ZAR image has 3 multi-resolution levels embedded in it, and it's giving me sort of a memory estimate of how much I'm going to have to download in order to view these and how much GPU memory it's going to consume. So this gives me a little hint of sort of what I'm in for if I select that level. So just for this demo, to try to be as efficient as possible, I'm going to load low-res data. Actually, I'm going to choose the middle resolution. Maybe I'll show one that's high-res at the end, but the rendering quality can sometimes hide whether the resolution is very good or not. This particular data set has 9 volume channels in it embedded. I'm going to just let them all load. That's fine. It also lets you choose a sub-region. So if you know you're only interested in a particular sub-volume of the data, you can choose that upfront and load even less data if you'd like to. For this data, it's fine. I'm going to just load the whole volume. We're ready to go. We click open. Okay, and then we get our image. So Agave is a progressive renderer, so the image is constantly refining and redrawing while I'm in the viewport. So when I start rotating using the same controls as I think Volley and Simularium Viewer, the image is very grainy and noisy. As soon as I stop, it refines and refines and refines. So it's sort of building up an average of many rendering passes. So what I'm seeing by default is the first channel in my data. The data channels are displayed in the lower portion of the left-hand panel. So I can turn channels on and off in my data set. This particular data set has segmentation data in it also, so I can see the computed shapes of cells, computed shapes of nuclei, and we also did a contour segmentation, so we can do these contour lines. One thing you'll notice is that even though this is segmentation data, it's a little bit cloudy and transparent. And that's an aspect of the software. So one of the first things that I would ever do when I load an image, this is my initial view that I see. The first control I would go to is scattering density. This is a big global control over the renderer that lets your data become more solid or more transparent, basically. So if, excuse me, if I have hard-edged objects that I'm trying to visualize, I would tend to turn the scattering density up. And if I think my data is has more of like a spread of intensities, then I might use a lower scattering density to reflect that. I'm going to turn on 2 channels of data right now. The next control that I would go to right after scattering density, actually, I'm going to isolate a single channel. The next control I would go to is the per channel thresholding and contrast adjustment controls. So I would go right into this, open up the channel, and we have a whole bunch of settings here. In particular, quickly, you can just change colors. We can go right to cyan if we want to. And then these two sliders are our threshold contrast adjustment controls. So I can move these sliders and sort of threshold out different parts of the data. There are a few different modes of usage here. There are 4 different ones. Each mode has a memory. So if I switch to the window and level mode, I have a different set of controls that I can use to threshold the data. And then I can go back to percentile mode and it remembers my percentile settings. So you kind of have 4 different views that will always be remembered here. The whole viewer, basically every setting in this viewer is meant to be played with and experimented with. They all do something to affect the appearance of your image. So we're trying to encourage you can play with this. But I'm just walking through sort of the most important key controls. In the custom mode, you actually are allowed to create, oh, sorry, I cannot do custom mode right now. I'm not going to show custom mode, but the idea is that we are, again, adjusting sliders to bring out intensities in our volume data that we want to look at. So really, like if I've done those 2 things, if I've dialed in a scattering density that looks good to me and a sort of contrast and thresholding setting, then I'm already kind of dialing in the appearance of my data. So the other thing, the very next thing I would do is change some of the lighting settings. So really simply, there are 2 light sources to deal with. And now at this point, you become a little bit of a lighting designer if you really want to control the appearance of the volumetric data. So there are 2 lights. One is a skylight, and the skylight is a completely spherical light source that's wrapping the entire volume. You get to adjust color and intensity of the skylight. And the skylight gets 3 controls for top, middle, and bottom. So the idea being that you're controlling the colors of a spherical light. And the top is sort of like the North Pole. The middle is like the equator, and the bottom is like the South Pole of that light. So if I just change the bottom light color to red, and I turn up the intensity quite a bit, you can start to see that there's a little bit of a red hue coming in only from one side of my volume data. I'm going to put that back to white and turn the intensity back down. And then the other light source we have is an area light. And the area light is a particularly directed light. So I can actually click on rotate. And now I get a rotation control. And it's kind of hard to see, but there are little lines coming out. I'm going to zoom out a little bit. If I zoom out, you can actually see this is the physical location of the light source with respect to the volume. And this all showed up just because I clicked rotate in area light. From here, I can just use this gizmo here and hover over the parts of this widget. And as I click and drag, it lets me move the light source. And it changes the direction that the light is impinging. So you can quickly drag this around. And it's hard to see now, but if I zoom in again, what we're doing here is we're changing basically the lighting and shadow direction on the data. I'm going to drag this to a different direction. And actually, I'm going to turn off the skylight by setting all its intensities to 0. Now the only light source on this is the area light. And as I move it, you can actually see the shadow direction change. So right now I've kind of dragged it so that it's coming from basically above, and the light is coming very much from a side angle onto my volume data. I can kind of rotate that all the way around. In fact, I can use this control to rotate it all the way around to the other direction. So this is the way you can play with the lighting directions to really affect the shadows that you see in the volume data. Why you might do this is basically 2 reasons. One is you want to bring out the spatial relationships in the data, and choosing the lighting angle can really highlight those spatial relationships. The other reason is you want to make a really beautiful image for publication or display. Okay, so I'm not going to touch every control because of limited time. I know I'm basically nearly out of time. Um, I'm going to click rotate again to hide the rotation controls for the light source. At this point, I can click this button up here called Quick Render, which will just save the current view as a PNG image file. If I have time series data, I can go into a special render dialog. This dialog box lets me change the output resolution, give me some basic image quality controls to render the same image that I just saw with higher quality. In this case, I have actually a low quality setting, but I can actually set a setting to say, I'm just going to let it iterate on the rendering for a fixed amount of time if I want to, or I can dial in a particular number of rendering samples. And then again, you can save the image out from this dialog box. This is the same dialog box that you would use for time series rendering too. It can iterate through an entire time sequence. The other things I will just mention about AGAVE is you can save the entire session. So all of the view settings that I've dialed in, every setting in the user interface with this Save to JSON button. For advanced users, you can also export all of these settings to a Python script, which that you can then use to drive AGAVE from Python. I'm going to leave that to reading the documentation and asking directed questions about it. That's a little bit more advanced use. I think I'm out of time for this. So I'm going to stop here and open it up to questions. I'm also going to load another data set of time series data just to show that. But yeah, that's about it.
Ooh, that was all. 6 tools.
Yeah, it's a lot.
Well, we have time for questions if anyone has any. Otherwise, we can totally leave it. Oh, we have a question. How would you say Agave compares to Blender?
To Blender? Oh, interesting. Agave is a very specialized tool for volume display in particular. Blender doesn't have direct loading of these types of biological image file formats. I'm pretty certain that Blender can't load Zeiss CZI microscope images. I don't think it can load ZAR data directly, so it would rely on plugins. Blender does give you a lot of these 3D controls and lighting controls, and probably even richer ones than what is in Agave, but it's a much more general purpose tool. Um, it also supports, hard surfaced geometries, right? It's probably more specialized for triangle mesh geometry data in general. It's also a fully featured animation and filmmaking toolkit, so it has a vast amount of more functionality than what Agave provides. But yeah, it's not specialized for biological image data, and I would say it's generally not specialized for this type of volume rendering, at least not in a live viewport like this.
Let us know if that doesn't answer your question. Oh, cool. We got to thank you. Any other questions?
Guys, absolutely fascinating tools. I would like to thank you for the demos and tutorials. Actually, I was working in Institute Pasteur and I came independently to this idea to move everything to the web, and it was a game changer for the lab. And I'm trying to build a startup around it right now. But my tools are really rudimentary compared to what you created. So congratulations on the great work, and I will definitely check them out.
Thank you.
Awesome. Yeah, we're happy to give additional demos or anything.
All right, I'm going to stop sharing and look at the chat.
Yeah. Leslie Barna says, very nice tools. Thank you. Pavlina says, I also applaud. I'm definitely going to try some of these out. I'm currently looking into the Timelapse Feature Explorer. I'm really excited whenever anyone's interested in working with TFE. So if you have any questions, my email's up above. We also have AICS software support. Um, any emails that you send there, we'll make sure they get to the right place. There's a GitHub repository also linked from TFE, and if you submit issue tickets there, that's a great way to let us know if there's a feature that you'd like. I believe most of our tools will have links to all of these are open source. So if you have questions, if you have issues, if you have stuff that you'd like to suggest or like to see, you can submit an issue ticket and we'll see that. So that's also a great way to get in touch with us.
Yeah, it's all open source. And also, we recognize that one of the challenging things can be getting your data in. We want to support that. So if you have questions about just how do I get my data into this tool, let any of us know and we'll try to help.
And it's great because once we know that it's a problem, we can make it easier for everyone else too. So we really appreciate it when people reach out. And then Pavlina, I like very briefly showed this off for TFE because it supports some local file loading. That's a good way to like if you're prototyping it and iterating on data, that'll make it like easier to you can just do stuff locally. But for all of the data sets that I shared that we were like the landing page links, those are all like hosted on HTTPS on like an S3 bucket. We recommend that if you're like working with a lot of different people because then it's easier to just like send a URL. You don't have to have people load local files every time. But at least if you're first getting started with it and you just want to prototype, there's lots of ways that to do that that will be faster.
Mhm. I think that might have been a follow-up to Dan's suggestion of maybe using something like an Nginx server to serve a network attached file storage for people like at core facilities that commonly have that.
All right.
Cool.
Uh, I think, oh, Sean, go for it.
I was just gonna say that's great. Thank you for listening, everyone.
Yeah. If you have any questions, feel free to stay on, but everyone else, thank you so much for coming.